Hello friends, welcome to BMH Learning. This video is regarding bile solubility test. The bile solubility test is used to identify and distinguish S. pneumoniae from alpha-lytic Streptococcus species. The test may be performed using a cell suspension on a slide or in a tube, or by adding the reagent directly to the colony. Pneumococcal cells are lysed when sodium disoxycholate is applied to the colony under specific conditions of time and temperature, but other streptococci do not lyse. The pneumococcus has an intracellular autolytic enzyme, an amidase, which causes the organism to undergo rapid autolysis when cultivated on an artificial medium. The bile salts alter the surface tension of the medium and cause cell membrane rearrangement. Media, reagents and supplies used. Reagents: bile salts. A 10% bile salt solution can be either purchased or prepared. Bile salt can be prepared by adding 10 grams of sodium disoxycholate in 100 ml of distilled water. The solution should then be dispensed in small amounts to minimize contamination. The shelf life of the solution is usually 270 days. The storage should be done at 15 to 30 degrees centigrade. The storage of the reagent at cold temperatures might result in the thickening of the reagent. 0.85% NaCl. Broth culture medium. You can use BHI. Procedure: bile solubility test can be performed either via the test tube method or direct plate method or direct slide blood culture method. Direct plate method. A drop of bile reagent is placed near a suspected 18 to 24-hour colony. The drop is then gently rolled over several representative colonies by tilting the plate. The colonies should not be dislodged with the bile reagent. The plate is kept right side up and is incubated at 35 degrees centigrade for 15 to 30 minutes. The plate can also be placed on a heat block as a substitute for the use of an incubator. The flattening of the colony is observed. Proper observation should be made to ensure that the colony does not simply float away. Direct slide blood culture test. 1 drop of blood culture broth is added to 1 drop of bile reagent on a glass slide and allowed to dry. As a control, 1 drop of broth blood culture is added to 1 drop of water and allowed to dry. The resulting suspension is gram stained and examined for cocci. Result interpretation. Direct slide blood culture method. If all the cocci in the smear are entirely lysed and the control smear shows intact bacteria, the organism is bile soluble. Direct plate method. Bile solubility is demonstrated as disintegration or flattening of the colony within 30 minutes, leaving an area of alpha hemolysis where the colonies are located. A negative result is demonstrated when there is no change in the integrity of colony within 30 minutes. This was all. Thanks for watching.